Good morning, everyone. Happy Friday. It's a little chilly here in Minneapolis, so I hope that you're in a warm place as you watch this today. My name is Ruby Nguyen, and first I want to thank you for those of you who attended last week when we had our joint seminar with Health Policy and Management. It went really well, I thought, and Health Policy and Management thought it went so well that they're already planning for us to do it again. And hopefully we'll be able to do that in the spring. If not, we'll be able to do it in the fall. Also slated is a duo seminar with biostatistics. So should you have any specific topics that you would like to have discussed with biostatistics, please do send me a note either in the chat today or directly to me on email. Can't believe it, but we're coming to a near end of the fall semester and our seminars. Let me just recap who is next. Next week will be Paul Robinson, who is a community leader and an outside consultant, and he'll be coming to speak with us about intercultural agility in workplaces. He's worked a lot with some of our faculty, and he'll be discussing some of the training programs that he has been a part of and has helped develop. And he's very excited about sharing some of that information with us. So we'll be welcoming him next week. The following week, of course, is Thanksgiving. And the week after that is an outside speaker, an epidemiologist coming in from Zooming in from Boston U. So she'll be speaking about, it'll be a surprise. I didn't memorize her title. I think I know, but I'm not going to go out on the ledge and be wrong. So we will actually drop our complete seminar series list in the chat box closer to the end of today's seminar. You can see exactly what Dr. Ellie Murray will be speaking about when she comes in from Boston U. So those are the next 3 weeks. And then we have 2 remaining seminars after that. Today we are welcoming one of our newest faculty members, Dr. Abigail Johnson. Dr. Johnson's research explores the relationships between diet and the human gut microbiome in health and disease using novel computational methods for dietary data. Her current focus areas include 1, analysis of diet and microbiome after dietary interventions, 2, methods development for dietary data analysis and visualization, and 3, understanding the interactions between foods, microbes, and fungi during different stages of development. She's interested in exploring how diet and the microbiome interact to influence chronic diseases, including prediabetes, diabetes, and cancer. Abby is a gopher through and through. She received her undergraduate degrees, dual. She double majored in nutrition and biology. And then also received her PhD in nutrition from this institution in 2015. Following that, she earned her RD degree. Abby joined us as faculty as an assistant professor, tenure track last year. So take it away, Abby. We're so excited to have you here as new faculty, but also have you here presenting in our seminar series. Thank you.
Okay, it's really nice to be here with everyone today. I hope that, like everyone always says, you can hear me and see my screen. It looks on my end that everything is okay.
Yes, it is, Abby. Thank you.
Perfect. Thank you. So today, and thank you for that kind introduction. And I'm very excited to be here with everyone today here. Nice to be on campus, although I do miss presenting to people face to face. Today, I'm going to be discussing some lessons learned and ideas that I have around integrating diet and microbiome data. And I will do that by telling some stories and vignettes from my research experience, and then end with a couple of recent examples from the literature that highlight the importance of some of this work. So this is one of my favorite images, and it's a false colored electron micrograph of the microbiome. So when I say microbiome, I'm talking about the bacteria, fungi, archaea, and their genomic potential, so the potential that they have to produce enzymes and proteins from their genes that reside inside of our bodies and on our bodies. Particularly, I am focused on the microbiome as it exists in the human gastrointestinal tract. So that's largely focused on the large intestine. So one of the reasons that I love this picture so much is that it shows a complex community, likely made up of both bacteria and fungi. You can see some fungal hyphae that extend throughout this image in the purple, and they are all interacting with a piece of incompletely digested plant fiber that at this magnification looks like a tree branch. So as a trained registered dietitian, this is amazing to me, just to see this in action, to see how bacteria are interacting with the foods or the byproducts of the foods that we consume. A good question when we start to think about the microbiome is, where does it come from? So this is a beautiful image that was created by Emily Lemming. She's at Twins UK, and I co-mentor her with Tim Spector. And Emily put this together to demonstrate that your microbiome really begins to be established from birth, right? Exposures like birth mode, whether you are born via vaginal birth or cesarean birth, are quickly, you then quickly accumulate more bacteria based on your exposures, early exposures like breastfeeding and formula feeding. And then as babies begin to interact with their environments, touching things, putting things in their mouth. If you've had a baby, you know that they're constantly doing hand to mouth motions as soon as they learn how to. There's this bloom of bacterial species that just continues to grow and change along the course of development over time. And as scientists interested not only in microbes, but in foods, it's really interesting to consider the ways that diet and microbiome, especially along this developmental trajectory, are inherently linked, right? These periods of rapid growth and development in which the microbiome is expanding correspond with periods of massive change in exposure to dietary substrates as infants become toddlers and explore food environments and eventually learn to make food choices of their own and develop likes and dislikes and eventually settle into a more stable adult eating pattern. So it's sort of obvious at this point, but it hasn't been obvious for a long time, that the microbiome adds this layer of complexity to how the foods that we consume affect our bodies and our health and well-being overall. So we've long understood that the foods that we consume are related to health outcomes, independent of microbiomes. So dietary intake is connected to weight gain and loss, blood sugar control, intestinal symptoms, and inflammation. But we now also understand that in addition to potential variation imparted by our human genome, there is also an enormous amount of variation that's imparted on these digestive processes based on the microbes that live in our gut. And those microbes, they potentially, so they metabolize the byproducts of our digestion. They probably also metabolize constitutively available substrates like the mucin that lines our gut barriers, like the small amounts of glucose and lactate that are produced by the enterocytes at the gut membrane. They also probably digest each other and their breakdown products, but they are awash in the foods that we consume. And so it makes sense that our diet shapes those communities in some way, shape, or form. And then the microbes themselves and their metabolites can then go on to influence our health outcomes. So it's really fascinating, and it's a really difficult set of processes to try to understand. The complexity in both diet and in the microbiome make both of these inputs really complicated. They're highly multivariate. And if we're going to understand them, we have to sort of start at the beginning, think really carefully about how we're going to do this research so that we're able to draw conclusions that we can trust. And so recently, with working with Uni Choi, she's an amazing postdoc in the Division of Epi and Community Health. Uni Choi and I have been pulling from the literature, exploring what's out there in terms of dietary pattern and microbiome, diet information. What's being done, how is this research being done? And what you're seeing here is a river plot or a stream diagram that shows you the results visually of our analysis of the literature. Basically, we found around 50 some research articles that have compared diet and overall dietary composition to the microbiome in some way, shape, or form. And the vast majority of that research is completed cross-sectionally, and it begins with food frequency questionnaires that are collected potentially many years prior to the microbiome outcomes of interest. And so what we're realizing is that the vast majority of the literature in this space, there's an abundance of data, but it has relatively limited resolution. There's a lot to be said about the ways that we could improve these processes and improve the quality of the literature overall. So one of the things that I was interested in exploring when I began my postdoc here at the U with Dan Knights back in 2016 was whether if we simply collect enough data at a high enough resolution, would we be able to identify how daily changes in our diet influence the microbiome within individuals? And so if you've never worked with microbiome data, what you're seeing here on this slide is what microbiome data looks like when we get it off a sequencer. This is actually my microbiome shown here in the background. So these are reads of genomic sequences that we get off of the sequencer that we then can take and computationally assign levels of taxonomy to. So for the study that I did as a postdoc, we collected microbiome data from 34 people. So this is a small, somewhat explorative study, but I think it still can tell us a lot of really interesting, or generate a lot of really interesting hypotheses. So we collected daily samples from 34 people, and those are shown here in stacked relative abundance plots. So the different colors here represent different species in the microbiomes of these individuals. And those species, you can see, vary a little bit from day to day over time. Each of these bars, is quite difficult to appreciate, is each person's 17-day time course. So if we expand one person's time course and make it wider so you can see it with a little bit more detail, you can see that the microbiome has points where it's measured each day of the study. And there's some variation over the course of the study with the microbiome composition of this individual moving around a little bit up and down, but generally the species composition stays largely the same. So we can visualize the microbiome in another way. So this is a principal coordinates plot or principal components plot, which shows each person's microbiome sample in a multidimensional space. Here, just showing the first 2 dimensions of that space. So principal coordinate 1, PC1, and principal coordinate 2, PC2. So to understand what you're seeing here, if you don't work with this type of data very often, each point is an individual's microbiome made up of all of the different species of that person's microbiome. And points that are most distant from one another in this space are more dissimilar. So the points over here of this individual in purple, their microbiome composition is highly different from the points over here of this person in lime green. So what we noticed was that lots of people had very different microbiomes, but they didn't change very much over time. That's the key takeaway point from this plot. So we also collected daily dietary records for the same time period. We asked individuals to tell us everything that they ate and drank using ASA24, which is an electronic tool for self-reported dietary recalls. It's available through the NCI. We asked people to collect their daily diet information for us for 17 days, corresponding to each of the microbiome samples. So the day before a person provided a microbiome sample, they kept track of everything that they ate and drank. And what you can see here on this figure, immediately, it should be obvious that there's a lot more variation. Those bars are filled with really spiky colors. They change a lot from day to day. And if I spread these out again, you can see that it's much more variable from day to day. But what's important to note here is that I'm showing you diet for this individual at sort of the highest, or I always get confused about this, highest or lowest resolution, but these are big food groups that I'm collapsing for visualization purposes. So the orange is grains, and the yellow or brownish color here is eggs, for example. As you can see, eggs are consumed on the weekend for this individual. And so you can see that there's a lot of variation in diet at this big food group level, whereas the microbiome, we see just a little bit of variation at the most specific level of species. If we tried to view diet in terms of individual food items that a person reported eating each day, we would be totally at a loss. It looks like a piece of Picasso art. There's almost no repetition. There are some things that this person consumes almost every day, but it's not consistent, right? Many people's diets are highly variable from day to day, which we know from previous work in nutritional epidemiology. I do want to go back and highlight, you may have noticed two individuals here who have been starred. Their daily intake is consistent, one thing throughout the entire study, and I will help explain that on the next slide. So when we visualize intake of individuals in this study in a different way using a network diagram, which is one of my favorite ways to visualize data to get an understanding of what's happening, here each person is a white dot, and each colorful dot on the periphery represents an individual food that was reported during the study, and each food is connected to a person if the person consumed it at any time during the study period. And so what you notice is that in the center of the plot, there are foods that were consumed by lots of people in the study. These are things that we come across frequently in the American diet, like white bread, iceberg lettuce, beer, wine, milk, tea, and coffee, items that make up the diets of many people habitually. On the periphery, you see things that are only consumed by maybe one or two people, and these were foods that were much more rare in our study. They were unusual organ meats, maybe some specific fruits or vegetables, for example. And I'll go back to my two individuals who just consumed one thing, which I think will help you understand how to interpret this visualization. Here, these dots are the two individuals who just consumed one or two things during the course of the study. This was an intervention study, or well, it wasn't an intervention, but that's aside the point. This was a study of students, actually a citizen science study. A lot of the participants in our study were students who were also taking a class learning how to study and analyze microbiome data. And what happened was that two of the students took it upon themselves to put themselves on a dietary protocol, consuming only a meal replacement beverage. So you can see the four different flavors of the meal replacement beverage that they consumed throughout the study, here represented in the purple, which is the sugar, sweets, and beverage color. And they're both tied into the network though, because on one day, one of them consumed a piece of pizza, and on another day, one of them consumed, they made a latte with their meal replacement beverage, so they actually consumed some coffee. If it weren't for those two foods that they consumed, these people would be off somewhere else in the network. So all of this to say that food intake is highly personalized, and it's really messy, and it's very difficult to deal with. Ordinarily, we manage some of this complexity by reducing the noise in our data by going and looking at nutrient composition. This allows us to explore variables that exist across all of the subjects, across all of the days. For example, we collapse this individual food information to protein, fat, carbohydrate, moisture, alcohol, for example. And depending on the database you use, you get between 50 to 200 nutrients that you can then assess. And so when we look at the nutrients for the participants in our study, indeed, these nutrients are more variable across the course of the study, with the most variability coming from nutrients like alcohol, which are consumed sporadically. And that's the sort of peach color that you can see popping up in these stacked relative abundance plots. And so back to the microbiome, what we aimed to do first was to ask whether or not each person's microbiome composition on average paired with their average nutrient composition. We wanted to know whether the nutrient composition of the diets of our participants was predictive, if you will, of their microbiome. And what we found was that the microbiome doesn't pair with nutrients. And this was fascinating. And I have a number of sort of hypotheses or theories as to why this is. One of them is that it's possible that by collapsing to nutrients, we are ignoring a lot of the other features of diet that exist at small quantities, but are potentially important from the perspective of your microbiome. So if you think about the flavonols, polyphenols, the different small components of foods that we don't capture in our nutrient databases because they are largely non-nutritive, they don't supply us with calories, and they are not considered essential nutrients from a human health perspective. These items of our foods potentially have an impact on the gut microbiome. So it might be useful to consider diet more holistically in ways that potentially capture some of this complexity without going through and making a database that's completely representative of every possible biochemical in existence. It's hypothesized or estimated that there's about 28,000 biochemicals in our food supply. So it's pretty unreasonable to think that we'll ever have a truly comprehensive nutritional database that really captures all of this complexity. Although some people are working to, throw every food through a mass spec to try to get more resolution. So the next question we asked was whether dietary pattern analysis can reveal important relationships with the microbiome. And I want to take a moment here to explain dietary pattern analysis and also plug the Nutrition Coordinating Center, where I am associate director under the guidance of Lisa Harnack, who's the director of NCC. So you can explore dietary patterns a number of ways. The first is using a priori, which are expert-defined dietary patterns. And here's my plug for NCC, is we are currently developing what should we call this? I'm blanking on what to call this. A healthy eating index report that will be embedded in NDSR. So hopefully it will be easy for researchers using NDSR in the future to get a healthy eating index, which is an a priori dietary pattern index for the people who are in their study, right? So a priori patterns ask whether or not individuals in your study are consuming a diet that aligns with a predefined dietary pattern. We want to know whether or not people are eating a specific way. So the benefit of doing an a priori diet pattern assessment is that you can take that information and compare it across other studies that also use the same index to ask whether or not the findings you found are similar to what other individuals have found in different studies. And so what we did in the study that I'm mostly sort of talking about today was we used an a posteriori approach, which takes a data-driven perspective. So a couple limitations with a data-driven dietary pattern approach is that these dietary patterns are going to be specific to the population from which they are developed. So we take the data for the specific population and ask which features of the diets of these individuals describe the most variation or capture the most variation in that population. So what we did for this diet microbiome study was we took the dietary information at the level of foods for each of the individuals. And what I noticed was that the FNDDS database, which ASA24 uses, is already organized in this hierarchical structure. And so we turned that hierarchical structure into a tree so that we could statistically account for the relatedness between foods. So we could take into account the food grouping structure when we asked whether or not a person's diet was more similar or dissimilar from one of the other members of the cohort. And what we found is that just like the microbiome, dietary information from our participants when we assessed it in this way showed that diet was individualized. So you can see that individuals here, while it's more variable, which we expect based on what we've already seen from this study, there are clusters for each individual. Like this individual colored in black, their diet is distinct and different from this individual colored in red, for example. And so diets analyzed in this way, we can explore and we can get distinct and variable dietary patterns for each of our individuals, just like we have distinct microbiome compositions for each individual. And then we can take the average of those. So just like we did with the nutrients, we can take the average microbiome information and their average food intake information, taking into account the relatedness of the foods that they consume. And we can ask whether or not those pair together. And what we found this time was that the microbiome paired with the habitual diet. Now, this is a Procrustes analysis. I don't think I've explained that fully on the previous slide. So here we're taking one set of points in multidimensional space and a second set of points in this multidimensional space, and we're twisting and turning them to see if those points correspond with one another more than what we'd expect if we shuffled them 1,000 times. And so in this case, we find agreement between these dietary patterns and their microbiome, which means that something about the dietary pattern is potentially informative about the microbiome composition. And so the other thing that this allows us to do, we've generated these tree-based dimensionality reduction features. And so what we can take now is each of these dietary patterns. So we can consider an axis in this space. These are orthogonal tree-based principal components that we can then consider to be dietary patterns, if you will. Each axis contains information about the intake of specific foods for each person. And so, for example, we found that the first axis of variation in our dietary pattern space was positively correlated with the intake of mixed grain meals and meats, and the second axis in this space was positively correlated with the intake of poultry and eggs and vegetables. And we can ask whether or not those dietary patterns correspond with species abundance in our sample. And what we found was that there indeed were relationships between these different dietary patterns and the microbiome compositions. And when we do a constrained ordination of our microbiome space by these dietary patterns, we find that it reveals a Bacteroidetes gradient that shows that there's higher abundance of phylum Bacteroidetes with the intake of some of these more, what you might consider westernized dietary patterns, or dietary patterns that are representative of kind of the American processed food diet. So higher intake of cakes and cheeses, for example, corresponding with higher intake of Bacteroidetes. But it's really important to note here that when we did this analysis, dietary patterns themselves explain approximately 45% of the variation in our microbiome distributions in this population. But if we just look at BMI, sex, and age, those combined explain 35%. And then if we combine diet, BMI, sex, and age, those combined explain 49%. So the amount of information in the dietary patterns themselves that is confounded by information about BMI, sex, and age is pretty substantial. And the actual amount of dietary pattern influence on the microbiome, independent of those things, is relatively small. The next thing that I want to talk about is the use of our longitudinal data. So I've talked at length about using our data on an average basis, so each individual's average microbiome. But the other thing that we did, because we had longitudinal data, was we were able to explore each person's variation in microbiome with their dietary variation over time. So here I'm showing how we take 17 days of microbiome data for one individual, 17 days of dietary data for that same individual, and ask whether or not those microbiome and diet principal components pair again with Procrustes analysis. And then for visualization purposes, I'm showing you the first component of that pairing along the axis of time. And you can see what happened in this study was that of the 34 participants, 28 of them actually had pairing significantly with their microbiome. And we estimate that diet explains about 25% of the variation within a person. What was really interesting is when we repeated this analysis, taking either 1 day of dietary history, trying to pair that with the microbiome, or 2 days of dietary history, trying to pair that with the microbiome, or 3 days, and so on and so forth, what we found was that the ability to pair with the microbiome increased with the number of days of dietary history that we took into account, with the best agreement when we took into account dietary history with a decaying influence. So taking 100% of the day prior's dietary information, 50% of the day before that's information, and 25% of the day before that's information, then almost all of the subjects have significant agreement with their microbiome. So that's sort of smoothing in the dietary space to remove some of that variability and error that's introduced because people's diets are so highly variable from day to day. Potentially improves the pairing with the microbiome. It's also, I think, important to note and interesting that we get pretty significant agreement when we take into account 3 days of dietary history. And this kind of aligns with what we know about transit time. Most people have a transit time, that is the time it takes for a food item that you consume to move through the entirety of the length of the gastrointestinal tract. That time is roughly 24 to 72 hours. So if you think about your gut as a bioreactor, it may make sense that food items that you consume today don't really have an impact on the variation in your gut microbiome for maybe 1 or 2 days, depending on how fast that motility through your gut, how long that process takes. And so this is a relatively interesting feature of diet microbiome analysis that I think deserves a little more attention. So I wrote a review recently with the goal of sort of taking the lessons that we've learned from looking at this dense diet and microbiome data to help to kind of design what I think would be the optimal study design for repeated sampling of diet and microbiome in an intervention, perhaps a dietary intervention or some other type of intervention. The first thing that I think is really important to incorporate is maybe a little bit implausible at the moment, but hopefully something that we can do in the future, is to stratify the sample by microbiome, or at least by habitual dietary features at the time of screening and enrollment, to help to account for some of the individual variability. So in my experience running a study, we found that by pure chance, our randomization study sample probably wasn't large enough, but the randomization ultimately resulted in one arm of the study having the majority of subjects who had a Prevotella dominant microbiome, while the other arm had a Bacteroides dominant microbiome. So when we tried to tease apart the influence of the intervention, it was totally obscured by the baseline differences in the participants in the microbiome. So best practice would be to be able to sequence the samples of your participants at screening or enrollment, and if not sequence them, perhaps to run some simple real-time PCRs to kind of assess whether or not they have key members of the microbiome at baseline, so that you can then stratify your samples accordingly to help to avoid some of that confounding by starting microbiome composition. Then I think the next thing that is potentially really important to do is to collect multiple consecutive microbiome samples at each of the time points of your study. This should help to remove some of the inherent variability in the microbiome by averaging repeated samples. And that has shown benefit in a number of studies now, helping to tease out actual signal from the noise. And then ideally, I think that collecting multiple diet records corresponding for each microbiome sample 2 to 3 days prior to each sample could potentially help to adjust for or account for dietary variation that is then incorporated into those microbiome samples. So that is kind of my ideal microbiome study design at this point. One of the key things that I've also discussed at length with collaborators is that we don't think standardization of the diet is appropriate in microbiome studies. So when I say that, it's often, discussed like, well, should we put our samples or our participants on the same diet prior to starting the intervention because we want to remove variability from their personal diet choices. But habitual diet seems to be important for shaping the microbiome. So if you suddenly shift all of the participants in your study to a new dietary pattern, you're adding variability that wasn't there before. So one idea that we think is potentially important to consider is this idea of stabilizing the diet around the time of microbiome collection. So how this might look is having your participants collect a record of what they consume in the 3 days prior to their microbiome sample, for example, and then asking those participants to use that record as a menu for their repeated samples in the future. So we're currently planning to test this idea. So with Ryan Demmer and Mark Pereira here in EpiCH, they are running the active gut study. So they have an R21 funded active gut study, which is an intervention purely interested in assessing the impact of exercise and walking on the microbiome. But what they've been kind enough to let me do is to test this idea of stabilized dietary control in the study. And what we are expecting we'll see is that some people in the study will adhere to their stabilized meal plan, and others in the study will deviate quite substantially. They'll be unable to follow their meal plan, and they will just eat whatever it is they were planning on eating originally. So hopefully we'll get a spectrum of people who are consuming their standard meal plan stabilized across their different microbiome collection time points. And we'll be able to compare that to the individuals who just ate their usual variability or usually variable diet to determine whether or not this stabilization actually reduces the variability in microbiome. And hopefully the next time I present to you all, I'll be able to tell you the results of that study. We've got about 15% or so of the participants enrolled, aiming for 100 people. So if you want to advertise our active gut study, please reach out and I will share the information with you. This is a study of individuals with prediabetes. The final thing I want to touch on today is kind of why this matters. Why should we be considering controlling for diet in the microbiome space? We could just randomize studies and the noise of diet will just fall out in the wash. So the microbiome space is filled with a lot of really compelling, really exciting, and really novel potential connections between the microbiome and health outcomes. One of the notable examples of this is with autism. So there have been a number of animal studies that have been suggestive of a causative role of the microbiome in outcomes in autism. And this has led to a lot of attention and focus in this area that affects real people who are really living with these conditions, real families, real children, real adults. And there's, a drive in some spaces to do things that are quite invasive, like fecal microbiota transplant to treat symptoms of autism, for example. And this paper actually came out yesterday, and it really highlights the importance of considering diet in these studies of microbiome. This study did an amazing job of collecting really detailed microbiome information in a large cohort, a large autism cohort that was studied for a long time in Australia, along with samples from their family members and their siblings who don't have autism. And they also collected, they're one of the first studies that also collected detailed dietary information and tried to look at the relationships between diet and the microbiome, with more of an attention to other dimensions of diet. So specifically, they look at not just nutritional composition of the diet, but also at the diversity of the diet and whether or not the diet was sort of high quality within a similar approach like the HEI, but for an Australian cohort. And what they find is that the relationships between autism and microbiome, decreased taxonomic diversity, and ultimately looser stool consistency are mediated by restrictive and repetitive traits, sensory anomalies in individuals with autism that ultimately are expressed as intake of decreased dietary diversity. So I'll bring it back around to this idea that I introduced early about kind of concurrent development of the microbiome and diet, but adding another layer here on of brain size. This is a figure that I put together for a preview for this paper that's going to be in Cell Metabolism, hopefully in the next couple of weeks. But basically, as the microbiome diversity of infants increases, we know dietary diversity is also increasing along that same trajectory, but the brain is also developing at the same time. So it's really important to not jump to brain microbiome connections when there's an important mediator like diet that's at play. So with that, I'll conclude that foods, diet patterns, and other dimensions of diet like diversity contribute in part to shaping the microbiome. Short-term dietary history contributes to individual microbiome variation, and controlling for diet using sophisticated methods for dietary pattern analysis may be especially important in observational studies that aim to connect the microbiome to health outcomes. So thank you to everyone who I've sort of mentioned, and hopefully along the way, with whom none of this research would have happened. So with that, I'll open to some questions.
Abigail, that was excellent. Thank you so very much. Excellent and exciting. I'm sure a lot of people have questions. What we'd like to do is ask you to use the raised hand function, and we'll call on you. If for some reason that doesn't work for you, please do type your question in the chat box, and Abby will be able to see it. Who would like to start? There's one from Jim. Would you like me to read it, Abby?
I can read it out loud if you want.
That'd be great.
Okay, so Jim says, beautiful slides, thank you. Can you give us a sense of the lab cost associated with sequencing one microbiome sample and analyzing repeated samples would be X times that amount? Yeah, so I didn't touch on it at length, but one of the key features of the study that I did with the repeated sampling was that we used shallow shotgun microbiome sequencing, which brings down the cost substantially. So with microbiome sequencing, especially metagenomic sequencing, which is not 16S, so most of the research that's done in big population studies tends to, or at least historically has been 16S sequencing, which takes a barcode of the microbe genome that has accumulated errors over time and uses that to identify who's there. Metagenomic sequencing sequences lots of little pieces of the microbial genomes and then kind of assembles that puzzle. And metagenomic sequencing, it's often been thought of that you have to go really deep, so like 7, 8, 10 million reads to get good resolution. And Dan Knights is kind of a pioneer in the space of demonstrating that shallower sequencing can drastically reduce the cost while retaining the sensitivity. So after a sort of 10,000, 20,000 reads, actually plateaus and you get pretty good assignment you can trust your assignments. So for that study that I reported on, we actually did what's considered very shallow shotgun sequencing with about 500,000 reads per sample, which got the cost down to roughly $50 a sample. So it was on par with 16S sequencing costs, but we were able to get species level resolution. At UMGC, depending on how many samples you put on a run, if you use the NovaSeq, you can kind of play with the cost, but a sequencing run will run you about $5,000, not including the extraction and library prep. But ultimately you can kind of get it down to about $50, $60 a sample, which is actually not as bad as you might anticipate. That was a really long answer. Hopefully I got your question answered.
You see Aaron's question listed there.
Yep, so I can read Aaron's question. So Aaron asks, says the biology is interesting, yet what do we know about whether manipulating microbiome can prevent any disease as opposed to being a consequence useful for treatment? Is this research likely to eventually alter diet recommendations? Yeah, I think that this is kind of the outstanding big question in the field, right? I haven't seen a lot of demonstration yet of manipulation of the microbiome with diet. It's often proposed as what we should do next based on all of the findings that we've got that these things are connected. But in practice, when people institute a dietary recommendation and test it across the population, they don't find big changes, or only a few people in their sample respond in the way that they were anticipating. So you see a lot of the research coming out with responder and non-responder, but much of it is not predefining what you think will determine if somebody responds. So it feels very cherry-picked. So I do think as we get more information, hopefully from this big Nutrition for Precision Health study, as well as what's being done with the PREDICT study and some of the commercial platforms that are doing this research like Zoe, hopefully we get more information. We can start to sort of narrow in on like, these are the features of a microbiome that we know will respond to an intervention that contains X fiber or X resistant starch, and therefore this person could benefit from consuming this food. I think that the most recent research on fermented foods has kind of been the most promising in terms of showing a conserved relationship across a population. But the people in that study consumed, I think, 6 servings of fermented foods a day to see a relatively small change in alpha diversity across everyone. So I think it will eventually lead to some altered diet recommendations, probably for clinical use before population level recommendations. But that's just my feeling right now.
Let's go to Diane's question next. Diane, would you like her to read it off, or would you like to ask visually?
I'm wondering about the interaction between stress and diet and the impact on the microbiome, and then on health outcomes, although based on your last question, you probably don't have an answer for the last part.
I don't know. My answer for that is I really don't know. I am not attuned to the stress diet microbiome literature, if there is any. I'm sure.
I mean, people talk about stress in the microbiome, and people talk
Right.
about dietary intake in the microbiome. So yeah, that's fine. That's
There's a lot of interest in this gut-brain axis. I would anticipate stress microbiome interactions have to be connected to the changes that people undergo, like in their diet when they're stressed as well. So yeah, I would suspect that a lot of the research might not have even looked at those relationships.
great talk, Abby. Thank you.
It really was, Abby. Let's go to David Jacobs next. Hi, David. How are you, David? As we wait for David, let's read a question out of the chat box. Can you comment further on the impact of BMI in the gut microbiome? It's from Carolina.
Yeah, there are some interesting relationships with where people consistently see with increasing BMI that microbiome diversity appears like it goes down. So whether or not that's caused by changes in physiology or bidirectional interactions between, say, immune status markers and the microbiome, I don't really know. I don't really know that we can comment on that. I would really like to see the world of microbiome embrace sort of a higher resolution of body composition assessment to explore more closely the relationships between like lean mass and microbe composition and other markers that might be more informative than simply BMI. But a lot of this early work in the microbiome space has been quite quickly and rapidly developed, I think, without a lot of thought to nuance involved in some of these measures. So I guess that's my comment on that. There likely are relationships. I think they're more complex than meets the eye at the surface view.
These are great questions. David, let's try again. Hi, David.
Okay. Hi. I'm sorry I was on mute and I couldn't get it off. What I was wondering is that it's very natural to recommend eating foods like yogurt or other probiotic foods. And I wonder if there's anything known about the effect of those on the microbiome, whether there is an effect, and if there is an effect, is it an effect that you would like?
Yeah, I mean, there's a long body of literature looking at fermented food, specifically yogurt intake, and their activity in the gut. Before microbiome sequencing was a thing, people have been studying whether or not specific strains or bacteria that are in yogurts show up in the gut, like if you plate the fecal sample and grow it, right? And I think what we see across the board is that a lot of the bacteria in those foods pass through the gut and show up transiently. This intervention that was conducted with, and now I'm blanking on the group, Christopher Gardner and the group out in California, they showed for the first time that intake of fermented foods, I think a lot of it was largely fermented dairy as well, but there were some fermented vegetables as well, was associated with increased alpha diversity, so increased gut diversity, which we generally think is a good thing, and with reduction in inflammatory markers across the board in all of the participants in their studies. So I suspect fermented foods are probably good, and that's been the messaging for a long time in the world of nutrition, especially with respect to yogurt. But we're just sort of getting more resolution to understand why that might be.
Thanks, Abby. Aaron has suggested a prospective study for you. Could you read his
Sure.
comment and then comment on it? Thank you.
Yeah, so Aaron says it's likely that different microbiomes are related differently to different diseases. It would be great to have a large enough prospective study of repeated microbiome measures with total mortality being the outcome. There are a huge number of confounders, though, beside diet, like medication. Yes, I agree. This would be really interesting. And I'm open to ideas if anyone wants to collaborate on launching such a study. There are ongoing studies like American Gut that have aimed to collect some of this data, but there hasn't been, and the Human Microbiome Project, which really focused on very, very, very healthy people, because there's still this question of what is a healthy microbiome that is largely unanswered. But I don't know of really large prospective studies of repeated measures that would ultimately link to a mortality outcome. Like we're talking then like a 30, 40-year study. Yeah, I don't know how to launch those sorts of things, but I'm open to suggestions if anyone knows how to do that kind of work.
Well, we've done it, but the question may be answered before that 30 or 40 years is up. You haven't commented on the willingness of people to collect samples And, that's been a challenge.
Yeah, actually, so I can comment a little bit. I mean, most of the people who've been in our studies have been really motivated, healthy, health conscious, interested participants, right? So that it's a different group of people than trying to get a representative population to collect stool samples. But the methods for stool sample collection, I think, are getting better and better. I've actually found that it's easier to get people to collect stool samples than it is for them to tell me their diet. I find that people react much more negatively to dietary collection protocols than they do to collecting a stool sample. So in my experience, once people have collected roughly three stool samples, they are okay with it and can do it. I do think that people who've had children and have changed diapers are generally more open to the idea than those who maybe have been a little bit insulated from having to interact with fecal matter in any way, shape, or form. But I generally think it's something that people will do, and the methods are getting easier to administer and then stomach for individuals. So,
Abby,
yeah.
Lisa and I have completely opposite questions, and mine was based on Erin. So let me ask that first, which was, how has it been transitioning into a unit in which we are just inundated by confounders, right? So we're not just looking at diet, we're looking at the whole structure. How has it been for you in this new world?
Good. I like I'm learning a lot, and I really appreciate the epidemiological way of thinking, and I'm sort of, I have a lot to learn, but I really do appreciate the complexity that I'm learning about, and I hope that it will kind of start to show up in my research moving forward. So yeah, I think it's exciting to sort of learn about all these other features and facets that others are interested in the division, because it helps to think about ways that we could be adding more layers of complexities to these discussions, right? Microbiome research is in its infancy and is at risk of repeating mistakes that other fields have, and missteps that other fields have made, right? Like there's been a lot of discussion recently, and it's really coming too late, about race and racism being perpetuated in microbiome research, and the sort of missteps of anthropology that are repeating themselves, if you will. So I think that having more voices in public health that can make sure that this complex lens of the way that we do our research and think critically about the research that we do, it's really important to bring to this space.
Great. So Lisa's question is about mammals who have many, many, many fewer confounders. So she wants to know more about the mouse models.
Yeah, I don't do mouse work. I really, like Lisa knows this, at an early stage, I sort of got out of the mouse world. But mouse models are fascinating, right? Because you can control a lot of things really tightly. There are also limitations in the mouse models and in how we've viewed their diets. I think that a lot of the way that we've treated chow diets versus like a high fat diet, for example, in mouse models has failed to account for the complexity of that chow diet. So chow diet is not a monolith that is, it's often made of byproducts of agriculture. Things that don't make their way into other places of the food stream might end up in a chow mouse diet. So I think that maybe we've underappreciated the diversity of dietary substrates in the chow mouse diets when we compare to like the high fat diets, which often have like a monolithic carbohydrate construction. So I don't know if that sort of gets at what you were thinking about, Lisa, but that's my gut reaction, if you will, that we, mouse models could be done better and fed more appropriately. And then, oftentimes maybe we should be thinking about mouse models as are they really repeats, or are they actually just the same thing? Mice are coprophagic, right? They eat each other's stool. So if you don't carefully account for that and keep them in different cages, you just have one replicate of your study. So lots to think about there.
So thank you again, Abby. Let me end on a comment from Nancy, which is, this was an excellent talk, Abby, and we are so glad that you are on our faculty. Welcome. So for the rest of you, we will see you next week. Take care. Have a good weekend and stay warm.